Here we go with the chromosomal basis of inheritance. So we've been looking at, we've already looked at mitosis, meiosis, how our chromosomes move around, get sorted into gametes, get passed on to zygotes, and we've looked at genetics and we're talking about kind of how that works. And what we're going to do now in chapter 15 is put the two of those together. The fact that the genes we look at in genetics are actually located on chromosomes and behave the way that follow the rules of chromosome movement and behavior. So, excuse me. Mitosis and meiosis were first described in the late 1800s, like around 1880. And Mendel was doing his work in the 1850s. So really kind of before they really knew what meiosis was. So according to the chromosome theory of inheritance, basically genes that Mendelian genes or genes that follow Mendelian genetics have specific loci, positions on chromosomes. And then chromosomes, as they move through meiosis, undergo segregation and independent assortment. And this agreed with Mendel's laws of segregation and independent assortment. Okay, so I kind of put it all together. Later on, this guy called Thomas Hunt Morgan was really the first person. It's like, Mendel described his stuff, then they talked about meiosis, and it seemed like it made sense and it fit. But the first kind of real evidence that genes were located on chromosomes came from this guy, Thomas Hunt Morgan. And Morgan did experiments with fruit flies, Drosophila melanogaster, D. melanogaster. So those are fruit flies. And he really provided evidence, experimental evidence that chromosomes are where Mendel's heritable factors are located. So Morgan chose fruit flies as his experimental organism because they're really a good model for studying genetics. Okay, as we've kind of figured out so far, studying genetics is really about looking about how traits are passed on to offspring. So if you want to study genetics, you have to study something that has a lot of offspring and that has offspring quickly. Okay, and fruit flies are kind of good at that. They have many, many offspring. They have a new generation every 2 weeks. And they also have a simple genetic makeup. Only 4 pairs of chromosomes. So their diploid number, 2n, is 8, and their haploid number is 4. Okay, and if you think about it, humans make a really, really crappy genetic organism or genetic model organism to study because we generally produce 1 or 2 offspring, and it takes 9 months for that offspring to be produced. So it's really, they use pedigrees to study matings and offspring that have already happened. But if you really want to look at studying human genetics, it's really kind of a tricky thing. So just some of the lingo that you'll see with the flies. Morgan called the wild type is the normal characteristic, and then traits. And those are the most common. Okay, so they are usually going to be dominant. Okay, traits that are alternative to the wild type are called mutant phenotypes. Okay, so you'll have the wild type and the mutant. And if you look here for flies at eye color, so red eyes were wild type and white eyes are mutant. So in one of Morgan's experiment, and this again helped look at the behavior of alleles with the behavior of chromosomes. So Morgan mated male flies that were mutant with female flies that were wild type. Okay, As would be expected, if you kind of had the red eye gene be dominant, the F1 generation all had red eyes. When they bred the F2 generation had a typical 3-to-1 phenotypic ratio, red eyes to white eyes, again, following normal laws of dominance, but only males had white eyes. And again, he probably produced 1,000 flies. Okay, and out of 1,000 flies, only males had white eyes. There were no females with white eyes in the F2 generation. So this was kind of fishy to him. And so basically what Morgan figured out is that this white-eyed mutant allele must be located on the X chromosome. It was the only thing that could explain that type of inheritance. Okay, and this finding supported the fact that alleles and genes have specific locations on specific chromosomes. So if we just kind of quickly look at the data, again, the parental generation, female wild type, male mutant, okay? F1 generation, all the offspring had red eyes regardless of sex. And then the F2 generation, all the females were wild type and only males had the mutant phenotype. So even though it was a normal phenotypic ratio, 3-to-1 for the F2 generation, it had that kind of unique, funny thing with only males having white eyes. And if you look at the chromosomes themselves, so again, you look on the X chromosome. So here, females are XX, males are XY. And you'll see here, the plus, this gene symbol, the plus means wild type, and without the plus is the mutant. And this is wild type. Okay. So if you sort out this into gametes, okay, the females can only put an X chromosome in the egg, so we don't need to do the whole box. Males can have either the X chromosome or the Y chromosome in the sperm. So, but again, with the dominant allele, all the offspring have a red allele, so, and the males only have a red allele. So they're all going to be wild type. And then if you cross a female from the F1 generation down here with a male from the F1 generation, okay, you're going to get, the female's going to be heterozygous for the allele, and the male is, they're always hemizygous, but it will give a wild type. So what you end up getting are red-eyed homozygous females, red-eyed heterozygous females, red-eyed males, and white-eyed males. Okay, so this kind of supports the fact that genes are in specific locations and specific chromosomes, and they move and get sorted, get segregated the way that the chromosomes do. Okay, that's it. And we're going to move on to look at linkage and sex linkage in the other screencasts.